Hello students, and welcome to video 2.4 on experimentation in psychology. This corresponds to your book sections 1.10 to 1.12 and also section 1.17. Alright, and so you learned about other descriptive studies in class in your reading, and so now we're about to go through the actual running of experiments in psychology, as opposed to descriptive studies that only describe the types of behaviors that we are observing. Experiments provide causal links between variables. So they're the only type of research that can say, yes, variable X causes this to happen to variable Y. So the major benefit is that they do provide a causal link between variables. And so while you're running your experiment, there are a couple steps that you need to make sure that you hit in order to properly run your experiment, and things that you should key in on as indicators of good science. So first, after you've decided what you're going to study, you need to select a sample group from the target population that you're going to study. And again, we use a representative sample of the population that we want to study so that we may have a large enough group of people or animals so that we can then generalize our results to the entire population we want to study. For example, if I wanted to run some kind of experiment on McKinley students, I would make sure I had a large enough number of students from each grade so that I could then, from my experiment, generalize my results to the entire McKinley student body. So now that I've decided on my target population, my experiment being McKinley student, and I've pulled a representative sample from the student body, I have to decide what my variables are going to be. So that depends on what I want to study. And so if I decide that I want to see if I can increase school spirit by increasing the school community feel, I have then decided on my independent and my dependent variables. And your independent variable is the thing that you manipulate. It's the thing that you change. And then from that, the dependent variable will change based on what you have done to the independent variable. And so again, you change the independent variable, and then based on that change, the dependent variable will also change. All right. So as you can see on this graphic here, the independent variable will force a change onto the dependent variable. After I get my independent variable, the thing that I'm going to change, and my dependent variable, what I'm going to measure, I have to then operationalize them. And by doing that, I come up with an operational definition, or a specific definition or identification of the steps that I'm going to use to control the experiment. In other words, it's a working definition of variables that my experiment is going to measure to be applied to the experiment. So in my example, by trying to increase school spirit by increasing the school community feel, I would need to operationalize that. I would need to come up with specific ways that I would test and measure those variables, because those terms are very general and they can mean a lot of different things. So I have to come up with specific ways that I will be measuring and testing them. And so for this experiment, I'm going to define school community, the thing that I change, as the number of pep rallies we have. Because I think that if we increase pep rallies, more people will be excited about going to McKinley or being a part of the McKinley student body or whatever. And my dependent variable, school spirit, I'm going to operationalize or come up with an operational definition of attendance at school sporting events. So again, I've gone from naming my independent variable as school community to an operational definition of the number of pep rallies that a student will attend. And I've gone from my dependent variable of school spirit, and I've come up with how I am going to be defining it for my experiment as attendance at school events. School sporting events. So you take the variables that you would like to determine a relationship between, and you outline specifically how you're going to measure and define them. Something that you need to be wary of when conducting experiments is the Hawthorne effect. And that is very similar to the observer effect, except it applies specifically to experiments. So you need to be sure that the behavior of your experiment participants isn't changing simply because they know that they're in an experiment. You have to make sure that the changes in behavior actually come from your changes in the independent variable and not simply because participants know that they're in an experiment. One way to do that is by grouping. You have your experimental control group versus your actual experimental group. And so you randomly assign one part of your participants to the control group, and they will get either no treatment whatsoever or a placebo. And I'll be explaining a placebo later on in the video. Or you assign them to the experimental group that does get that change in the independent variable. And so this is so that you can make sure that the changes in behavior are a result of the independent variable and not because of that Hawthorne effect. Again, you have to make sure that you randomly assign participants to these groups so that you can make sure that you're minimizing the effect of those outside variables. For my experiment example of trying to increase school spirit, I have to make sure that I'm randomly assigning students to either the control or the experimental group so I can try to limit the natural presence of school spirit. Maybe some students are just naturally more spirited than others in my study, and so I have to make sure that I'm minimizing that effect. Randomly assigning your participants to their groups minimizes the effect of outside variables. Don't confuse this with random selection. Random selection is how you at first pull people into your study. And then after you have your sample pool, you then randomly assign them to either the control or the experimental group. As with all other studies we've looked at, there are some things that you have to be careful for. First being the Hawthorne effect, which we've already discussed, and the next two being the placebo effect and the experimenter effect. Now, the placebo effect is when a person's belief that something will happen as a result in the experiment affects the results of the study. For example, if I was testing a new drug that is supposed to improve memory, some participants would see an effect on their memory simply because they believe that whatever pill they're taking is supposed to improve their memory. So in other words, a person's expectations or biases affect their behavior. If you're expecting an effect by being in a certain experiment, then it's likely that you will see that effect. You see this more often when you test things like drugs, because you will give participants what's called a placebo or a sugar pill or a salt pill, just so they don't know for a fact if they're getting the drug or not. So in these kinds of studies, the control group will see a modest amount of change simply because of this placebo effect. People expect a change, so they see one. Something else you have to look out for is the experimenter effect. So whether intentionally or unintentionally, the experimenter can indicate the proper response to what is happening through the independent variable. So going back to my specific example, I can accidentally or on purpose suggest to the people who I have attend more pep rallies to go to more sporting events. Knowing that you're in my study, I could ask you, so are you going to any of the sporting events this weekend or after school or so on and so forth? And through knowing that I'm the experimenter and that I'm testing something, you could be clued in to the fact that you're supposed to be going to more sporting events. And so you would do that because I have clued you in and not because my experiment has worked. And so there are a couple of ways that you would control for these hazards, and they are single blind studies in which the participants don't know which group they're in, control or experimental, and they are double blind studies in which neither the participants nor the researcher know which group they're in. And so for a single blind, if you're testing a drug, the participant doesn't know if they're getting the control pill, the sugar or salt pill that does nothing, or the actual pill that we want to see if has an effect, that experimental pill. And in a double blind study, neither the experimenter nor the participant know what group they're in, so they don't know if they have that sugar pill or the actual thing with the medicine in it. This just limits both the placebo effect by having the participant be blinded and the experimenter effect by having both the participant and the researcher be blind to which group that person is assigned to. And now that we've covered descriptive statistics and we've covered experiments, we can now talk about inferential statistics. And this is what we as researchers use to make inferences or draw conclusions based on our data. This is how we make sure that we can say that yes, this X thing causes this Y response to happen. Also, running inferential statistics lets us know that we haven't just gotten our results by chance. We can say through running these tests that it is unlikely that we got the change between the control and the experimental group by chance. And so these inferential statistics allows us to make a comment on our statistical significance. And so it is just a way to test the differences in our groups to ensure that they are real and that they are not caused by outside variables or by chance. And so when you hear people say that this result is statistically significant, it's another way of saying that the probability that we got this result is less than 5%. We'll discuss more of these topics in class. Hope you take great notes. Again, write any questions you have down and we can unpack them in class.